Good morning, students. Today we are going to discussing about very important another 5 marks question in PUC examination, that is also known as double fertilization. Number of times in PUC examination directly diagram is asked. In sometimes diagram along with the explanation also asked in PUC examination. First of all, what is the definitions of double fertilization? Here the fusion of two male gametes with two different components of embryo sac, namely egg and secondary nucleus, is called double fertilization. Generally, two male gametes are released into embryo sac. The two male gametes are fuses with two different components of embryo sac, such as egg and secondary nucleus, that is called double fertilization. Here, this is one type of the definition. One more definition. The whole process of fertilization that involves syngamy and triple fusion is called double fertilization. Here, later we will discuss the syngamy and triple fusion. The whole process of fertilization that involves syngamy and triple fusion is called double fertilization. One more definition. The occurrence of syngamy and triple fusion in an embryo sac of angiosperms is called double fertilization. If you write any definition in examination, compulsory give the mark. That's why main important point of double fertilization is, in that double fertilization, two different types of the fertilizations are involved, that is syngamy and triple fusion. That's why the occurrence of syngamy and triple fusion in an embryo sac of angiosperms is called double fertilization. This is definitions of double fertilization. You know that embryo sac. Generally, the double fertilization mainly takes place inside of the embryo sac. How to double fertilization occurred inside of the embryo sac? First of all, the pollen tube. This is nothing but a pollen tube. Generally, this is actually stigma. This is a style. This is ovary. Inside of the ovary, this is ovule. Inside of the ovule, this is embryo sac. This is ovary. This is ovule. This is a embryo sac. Here, first of all, the pollen grain fall on the stigma. From that pollen grain, pollen tube germinated any one of the germopore. The pollen tube transported through the tissue of stigma and tissue of style and finally entered into ovule. Later, here finally the pollen tube is entered into inside of the embryo sac. That's why actually inside of the pollen tube two male gametes are present. Inside of the pollen tube two male gametes are present. Here you know that how do the two male gametes are developed from the pollen grain. You know that the entire aspect. Leave it that one. First of all, the pollen tube carrying two haploid male gametes and entered into embryo sac. Any one of the synergids. Here actually two synergids are present in embryo sac. The pollen tube entered into embryo sac. Any one of the synergids. Here, first of all, how to pollen tube transported towards to embryo sac. In that condition, generally, this is nothing but micropylar end. This is the chalagen end. Actually, here towards the micropylar end, egg and two synergids are present. The egg and two synergids are combined together to form egg apparatus. Generally, either side of the egg, two synergids are present. The synergids have a special cellular thickening. Finger-like projection is also known as a filiform apparatus. From that filiform apparatus, a specific type of the chemicals are released. That chemicals are mainly helpful for guide the pollen tube towards embryo sac. How to pollen tube entered inside of the embryo sac. For that entry of the pollen tube, here which part of the embryo sacs are helpful? Which part of the embryo sacs are mainly helpful? Here that is also nothing but synergids. Actually, from that piliform apparatus, a specific type of the chemicals are released. That chemicals are mainly helpful for guide the entry of pollen tube towards the embryo sac. This type of the movement is also known as chemotropic movement. Next one. Here, any one of the synergy degenerating when the pollen tube comes near to the egg apparatus. Any one of the synergy degenerates when the pollen tube comes near to the egg apparatus. The pollen tube entered into embryo sac through degenerating synergid. Here, the pollen tube is entered. Next one. Another one, next one that is also known as when the tip of the pollen tube entered into embryo sac, the vegetative nucleus or tube nucleus are degenerate. Especially inside of the pollen tube, one vegetative cell and two male gametes are present. Once the pollen tube entered into inside of the embryo sac, here vegetative cells are also degenerate inside of the pollen tube. Next one. Here the tip of the pollen tube is swelling. The tip of the pollen tube is swelling or burst after enter the inside of the embryo sac. Here the pollen tube entered inside of the embryo sac. The tip of the pollen tube is bursted or swelling after enter the inside of the embryo sac. In that condition, two male gametes are released into inside of the cytoplasm of the synergids. The two male gametes are released into inside of the cytoplasm of the synergid. In that condition, here two dark granules appears in the regions of degenerating synergy. Two dark granules are appears in the regions of degenerating synergies. These granules are nothing but X bodies. X bodies. These X bodies are mainly helpful for degenerating the tube nucleus as well as synergid nucleus. In this way, tube nucleus and synergid nucleus are disappears inside of the embryo sac. In this way, two male gametes are released into inside of the embryo sac. Later, here the two male gametes are fuses with two different components of embryo sac. That is nothing but double fertilization. How do two male gametes are fuses with two different components of embryo sac? Now we are see that aspect. First of all, here during that. Here are explanations of definitions. During the explanations of definition, already what is the definitions of double fertilization? The whole process of fertilization that involves in syngamy and triple fusion, that is also known as double fertilization. First of all, syngamy. What is meant by syngamy? If you clearly observe the diagram, totally how many number of male gametes are released into inside of the embryo sac? Two male gametes. This is the first male gamete. This is the second male gamete. The male gamete ploid is haploid. The first male gamete fuses with egg. To form diploid zygote. Here, this is a one male gamete. N fuses with egg N to form diploid zygote. Here actually the ploidy of the zygote is 2N. This process is also known as syngamy. One more name of the syngamy: real fertilization, true fertilization, generative fertilization, and one more name is sexual fertilization. Here, sexual fertilization. One male gamete fuses with the egg to form diploid zygote. That is called syngamy. That is called real fertilization. That is called true fertilization. That is called generative fertilization. That is also sexual fertilization. This is nothing but syngamy. It was first discovered by Strasburger in Monotropa. It was first discovered by Strasburger in Monotropa. This is also this is nothing but a syngamy. Very simple answer. What is meant by syngamy? One male gamete fuses with the egg cell to form diploid zygote. That is called syngamy. That is called true fertilization, generative fertilization, real fertilization, sexual fertilization. It was first discovered by Strasburger in Monotropa. Second one is triple fusion. What is meant by triple fusion? The second male gamete fuses with secondary nucleus in the central cell. Is called triple fusion. Here, second male gamete N fuses with secondary nucleus 2N to form triploid primary endosperm nucleus. The second male gamete fuses with the secondary nucleus to form triploid primary endosperm nucleus. How to develop the triploid primary endosperm nucleus? Here, triploid primary endosperm nucleus is a resultant structure of triple fusion. How many number of nucleus are involved in triple fusion? One male gamete nucleus, two secondary nucleus, two nucleus of secondary nucleus. That's why totally n plus 2n, 3n form of product is formed. This is nothing but a triple fusion. This is nothing but a triple fusion. Here one more name of the triple fusion is vegetative fertilization. Vegetative fertilization. Vegetative fertilization. The second male gamete fuses with the secondary nucleus to form triploid primary endosperm nucleus. It is called triple fusion. It is called vegetative fertilization. It was first discovered by Navasin in Lilium and Fritillaria. This is also known as triple fusion. What is the definitions of double fertilization? Here the occurrence of syngamy and triple fusion in an embryo sac of angiosperm is called double fertilization. Syngamy and triple fusion, both type of fertilizations takes place in only inside of the embryo sac. That's why the occurrence of syngamy and triple fusion in an embryo sac of angiosperm is called double fertilization. Here, since the two types of the fusions such as syngamy and triple fusion takes place in inside of the embryo sac, this phenomenon is called double fertilization. This is unique features among all flowering plants. Double fertilization is a unique features among all flowering plant like angiosperms. Here actually the double fertilization is completely absent in gymnosperms. Generally in gymnosperms triple fusion is absent. That's why in gymnosperms generally the product of triple fusion is endosperm. The product of triple fusion is endosperm. The ploidy of the endosperm is 3n. In all angiospermic plant, the product of triple fusion is endosperm. Endosperm supplies the food material for the development of the embryo. That type of the endosperm is not developed in gymnospermic plants. In gymnosperms, female gametophyte act as a endosperm. Embryo sac is female gametophyte is act as a endosperm. Why female gametophyte act as endosperm in gymnosperms? Because of in gymnosperms triple fusion is absent. Due to the absence of triple fusion in gymnosperms, female gametophyte is act as endosperm. But in angiosperms, triple fusion is present. Due to the presence of triple fusion, endosperm is present. Endosperm is formed. The endosperm is act as a nutritive tissue. Next, one more point. Here, double fertilization and triple fusion. Triple fusion is a specialized overall universal characters in all angiospermic plants. Here, the central cell after triple fusion. This is actually central cell. The central cell after triple fusion, became triploid primary endosperm nucleus. After that, the triploid primary endosperm nucleus is developed into endosperm. Here, endosperm is a product of triple fusion. Embryo is a product of syngamy. Later, the zygote is converted into embryo. Zygote is converted into embryo. Here, embryo is a product of syngamy. That's why two types of fertilization takes place inside of the embryo sac. Later, all remaining cells of embryo sac, such as antipodals, synergids, degenerating, excluding zygote and secondary nucleus after fertilization. After complete the syngamy and triple fusion, except zygote and secondary nucleus, except zygote and endosperm, remaining all embryo sac cells are completely degenerated. This is also known as double fertilization. Here actually, which type of the questions are mostly asked in NEET examination? Here, how many genomes are involved in double fertilization? First of all, what is meant by genome? The haploid set of chromosome is called genome. Otherwise, one nucleus is act as a one genome. Here, egg is one genome. Two male gametes are two genomes, and the secondary nucleus is two genomes. Totally, how many number of genomes are involved in double fertilization? 5. 1, 2, 3, 4, 5. How many number of genomes of embryo sacs are involved in double fertilization? 3. How many number of genomes of embryo sacs are involved in double fertilization? 3. 1, 2, 3. How many number of male gametes genomes are involved in double fertilization? 2. 1, 2. Next one. How many nucleus are involved in double fertilization? Totally 5. How many gametes are involved in double fertilization? 2 gametes. How many gametes are involved in double fertilization? Totally 3. 2 male gametes, 1 female gamete. Here, triple fusion mainly occurs in which place of the embryo cell? Exactly in central cell position. Totally, how many genomes are degenerate after complete the double fertilization? Totally 5. 2 synergid genomes, 3 antipodals genomes. How many genomes are degenerate after double fertilization? Totally 5. How many genomes are degenerate towards micropylary end? 2 genomes. How many genomes are degenerate towards charger end? 3 genomes. Totally here, the largest cell of the embryo sac is central cell. The second largest cell of the embryo sac is egg. Very smallest cells of the embryo sac is antipodals. This is also known as here double fertilization total information. Thank you all of you.